Welcome and thank you for standing by. At this time, all participant lines on the phone are in listen-only mode. After today's presentation, you'll have the opportunity to ask questions, and you may do so at that time on the phone by pressing star, then 1. Today's conference call is now being recorded. If you have any objections to this, please disconnect at this time. And now I will turn the call over to your host for today, Dr. Sevgi Aral.
Thank you. Thank you, everyone, for being here, and good afternoon. I can't tell you just how pleased I am to introduce to you Dr. Sheila Lukehart. I know she's not a stranger to any of you. She's, in fact, a really close friend of ours in the division. Sheila comes to us from the University of Washington, where she's a professor of medicine and global health, and Adjunct Professor of Microbiology, as well as the Director of the STD/AIDS Research Training Center at the University of Washington. I think sometimes it's difficult to describe people. I think it's really easy to describe Sheila. She is the leading syphilis and pallidum researcher in the country. That's really clear. She received the ASTDA Distinguished Career Award, which used to be known as the Parent Award, in 2007, and her research has focused on molecular pathogenesis of syphilis, on immune responses to and antibiotic resistance in T. pallidum, on neurosyphilis, and on syphilis-HIV interactions. Of course, we are all grateful to both the ASTDA and CDC, who are the co-sponsors of the STD Prevention Science Series lectures, and this is the second 2016 STD Prevention Science Series lecture. Without much ado, thank you for being here, Sheila, and it's all yours.
Well, thank you very much, Sevgi. That was a very kind introduction, and thank you to ASTDA and CDC for having me. I told Sevgi yesterday that I always love to come to CDC. It's such an interesting and diverse place. It's a wonderful place to be. Well, what I'd like to talk to you about today is my favorite disease, which my mother would never tell anyone. I want to talk to you about syphilis, and as Sevgi said, I've spent my career trying to understand how the bacterium and the host interact with each other to cause this incredibly fascinating disease. I call it a complex disease, and I'll try to tell you a little bit about why it's complex. And as I go along, if you have any questions, if there's something you don't understand, please let me know, because if you don't understand, others may not as well, and I want to explain things as I go. I know this is not a group of basic scientists. There are some of you in the group, but not everyone, and so I'm going to try to describe some basic science concepts to you so that the language won't get in your way. That's my goal. So it's just the language. The concepts are pretty easy. So I said that syphilis is a complex disease. It's complex from various points of view. Epidemiologically, it's complex. It shifts from one population to another over time. And it's a disease that may be seen in the setting of other diseases, such as HIV, or other infections, such as HIV, which may result in immunosuppression. And the other interesting thing about syphilis, which differs from many other infections like measles, mumps, et cetera, is that reinfection is common. You can be reinfected once you've had syphilis. So it makes it a challenging disease to study. And I know this is bringing coals to Newcastle. You're the ones who actually came up with these data, but just for those of you who don't know the syphilis data, syphilis, there's about 20,000 cases reported in 2014 of primary and secondary syphilis. And those stages I'll describe to you in a minute. The reason that's looked at separately is because those are the stages of syphilis that are infectious. And so it's kind of a marker for how much syphilis is moving through the community. And that's up 15% over the previous year. So this is an infection that's undergoing a huge increase. All stages of syphilis up 12%, about 63,000 cases, and congenital syphilis, which is when the organism crosses the placenta in a pregnant woman to infect the fetus in utero, and can have very, very severe sequelae was up 27.5% over the 2013 numbers to 458 cases. And right now in the United States, 83% of primary and secondary syphilis is seen in men who have sex with men. 51% of those are HIV infected, so co-infection is quite common. This shows you the number. I'm supposed to point with a pointer, so I'll do this. This shows you the number of syphilis cases reported in the United States. It's on a logarithmic scale. I want you to notice that. So from here to here is a tenfold increase in cases from 1940 to 2014. And there are three periods of time that I want to emphasize at the end. But just to start out at the beginning, in the first part of the 20th century and the last part of the 19th century, syphilis was an incredibly common disease. It was a disease that affected probably 10%, 5 to 10, maybe 15% of the population, both in the United States and in Europe. It was very, very common. And because it can cause really serious late sequelae, it was seen as a huge burden. And Thomas Perrin, for whom the ASTDA award was originally named, instituted what he called the syphilis dragnet, which was a mechanism for identifying cases, contact tracing, and going out and treating those individuals. Because at the time that he was Surgeon General of the United States, which was in the late 30s, early 40s, they finally had a good treatment for syphilis, which was penicillin. They had serological tests, and they had for diagnosis, so they could do the case identification that they needed to do. And as you can see right here, in the late 1940s, this is when the war was over, and penicillin was able to be made available to the population, to the general population, not just for military uses. And the syphilis dragnet reduced the number of primary and secondary syphilis cases to an incredible low. So this was the 50s, and Father Knows Best was on TV, and everything was good, and everything was wonderful, and syphilis was gone, is what people thought. And syphilis funding for syphilis research was decreased tremendously. And you can see that after that time, if I can get the pointer back, syphilis came back, and it underwent these about every 10-year cycles of increase and decline. Well, that's changed in recent times, and I want to just point out these three times. In the early 1980s, syphilis was found again in MSM, men who had sex with men. And it was during this time that HIV was also identified, and there were behavioral changes that occurred in the MSM population, use of condoms, et cetera, that caused a decline in syphilis in that population. And then in the mid-80s, peaking in 1990, there was another episode of syphilis in the United States, another epidemic of syphilis in the U.S., and this was largely in African Americans, largely in inner cities, but not completely. And it was a heterosexual epidemic. And then that epidemic went away. We reached a point in 2000 where it was nearly as low. It was actually a little bit lower, than it was in the mid-50s. And then syphilis began to increase again. So I'm going to give you some details about these two periods of time. You can see that the 1990 epidemic was very clearly a heterosexual epidemic, found in both men and women. And then the epidemic that began in 2000, and I use the word epidemic probably not correctly because I'm not an epidemiologist, but you know what I mean, spreading widely. This is almost exclusively in men, and the male-to-female ratio in 2014 was nearly 10. So this is largely in men who have sex with men. And then if you look at race ethnicity, you can see, again, that the 1990 epidemic was largely in African Americans, and the epidemic now is quite mixed in terms of African American, Caucasian, and Hispanic. Other groups affected less so. I'm going to describe syphilis to you very quickly for those of you who don't know. This is an infection. It's sexually transmitted infection. It is caused by direct contact with an infectious lesion, so that's why people with primary and secondary syphilis are the infectious persons. And the organism gets through the either abraded skin or mucosal membranes, and it begins to multiply locally. And after a period anywhere from 2 to 6 weeks, you see the first clinical manifestation, which is shown here. That's the primary chancre. It's an ulcerative lesion, has literally millions of treponemes on the surface, so readily transmitted, and billions of treponemes in the tissue right surrounding this particular chancre. And then the immune response comes in and clears that primary chancre. It heals spontaneously. Patient may think he or she is completely cured, and then nothing happens for a while. This period of time is generally, the time between primary and secondary syphilis is generally 3 to 4 weeks or even several months before development of the secondary stage, which is usually seen as a skin rash. And this rash can be of almost any type, begins as flat macules, then these lesions enlarge and become raised, so it becomes papular, which is what you see here. And then they may become vesicular, or may become pustular, which is the latest version or the last version of this particular stage of the infection. But again, the host immune response comes in, clears literally billions of organisms from these sites, and the infection apparently goes away. It doesn't really go away. It has traveled to other parts of the body, and individuals who have syphilis remain infected for the rest of their lives unless they're treated with penicillin and appropriate antibiotics. And in the pre-antibiotic era, about 70% of people stayed latently infected for the rest of their lives, and they died of something else. 30% of people developed either gummatous, cardiovascular, or symptomatic neurosyphilis, symptomatic late neurosyphilis, as you see here, and those are called the tertiary stages. But what's left off of this diagram, and which I think is being rediscovered, has been rediscovered in about the last 20 years, is that very, very early in infection, this organism invades the bloodstream and to the lymphatics and spreads throughout the body. And this can happen even before the development of the primary chancre. So it's a very early event. And obviously, dissemination of the organism is necessary to have the rash of secondary syphilis. It's necessary to have people with treponemes in different parts of their body during latency. It's certainly necessary to reach any of these tertiary manifestations. The other thing that it does is it goes into the central nervous system, as I mentioned, and it also crosses the placenta to cause congenital syphilis. But I think the earliness and the extent of this invasion and dissemination for many years was forgotten, and we only thought about this part of the natural history. So it's a complex disease biologically as well. I just showed you it happens in discrete clinical stages. The early lesions heal completely, but the infection persists. So this is not something that your body cures itself of. It disseminates to nearly every organ. It gets into the fluid in the eye. It can get into the fluid in the ear, crosses the placenta, gets into the central nervous system, and stays in these areas for a lifetime. It can also reappear after decades. The tertiary manifestations may not appear for 20, 30, 40 years after initial infection. And then again, this recurrent theme, syphilis reinfection, can occur. So there's not a protective immunity that develops during the course of human syphilis. It's certainly not early human syphilis. So who causes this? Well, Treponema pallidum, subspecies pallidum, the organism. It's shown right here by a silver stain. You can see these spiral-shaped organisms. This is probably a primary chancre. There are a lot of spirochetes here. This is a cryo-EM computer rendering of Treponema pallidum. It's a gram-negative organism, but it's very different from most gram-negative organisms in that it doesn't have lipopolysaccharide, but it does have the outer membrane, then the cell wall, then the inner membrane. You can see right here the outer membrane. Down here is the cell wall. Here is the cell membrane, and this is the inside of the bacteria. And in between the outer membrane and the cell wall are the flagella. Now, typically in bacteria, flagella kind of hang out outside of the body, and they flap around, and that's what makes the organism move. In this bug, the flagella are wrapped around the body underneath the outer membrane. So the outer membrane's gone in this rendering so that you can see the endoflagella. But they rotate internally, and that causes the organism to move in its typical spiral shape. So this is an odd bug in that it can't be grown in vitro, which makes it quite difficult to work on. It has a very, very small genome, and what it has done through evolution is that it has basically gotten rid of all of its metabolic capabilities. For those of you who remember your biochemistry, it has glycolysis, which is one way of making energy from sugar, but it doesn't go through the next steps, which really amplifies the amount of energy that can be made, so it grows very, very slowly. It can't make its own proteins. It can't make its own lipids, things that other bacteria typically can do. And consequently, it's left with its genome only with those characteristics that it really needs to survive as an organism. So I told you that early in infection, there's invasion and dissemination. And the key to dissemination is getting into the bloodstream. First, you get through the skin, but you also have to get into the bloodstream. And the capillaries are the place that this happens. And capillaries are lined by a single layer of cells called endothelial cells, and they're stuck together with what are called tight junctions, such that the blood can't leak out, and theoretically other things can't get in. So this is what keeps your circulatory system intact. And what happens with T. pallidum is that it migrates between these endothelial cells. I think you can see it here. This is a cell. This is another cell. This is the space between the cells, and you can see the dark spirochetes that are migrating through these cells. And recently, Caroline Cameron, a fabulous investigator at the University of Victoria, has discovered that a particular protein, TP0751, is important in binding and degrading laminin. So what's laminin and how does that fit in? So say this is your skin, and your skin really has multiple layers of epithelial cells, but this is a single layer shown in this cartoon. Underneath that is the basement membrane, and the basement membrane is made mostly of laminin. Similarly, this is a capillary, or a blood vessel, and the endothelial cells, those cells that are tightly pushed together, are this red line, and then the pink line just outside of that, again, is another basement membrane, and it has laminin in it. So what T. pallidum does, and this is a slide that Caroline gave me permission to use, is that this spirochete will bind to something called extracellular matrix on the surface of cells. And extracellular matrix is a combination of different proteins that kind of make, I think of kind of cotton wool. They're kind of filamentous and they're sticky, and things will bind to them. And T. pallidum does have some proteins that will facilitate that binding to this extracellular matrix. And then T. pallidum also has proteins that will bind it more tightly to the host cell. And this is the host endothelium. You can see here that there's a nice tight junction here. And TP0751, you can see here in this blow-up of this particular section. This is the treponemal membrane, the bacterial membrane, and so inserted in that membrane is a lipid or a fatty acid tail that holds this protein in close proximity to the organism. This is 0751, and it binds to the ECM on the surface of the endothelial cell. This protein, 0751, also cuts itself. It's a protease, so it cuts proteins, and it cuts off part of itself and changes structure. As you can see here, it's gone from this structure to this structure. And this structure now can bind closely to the host cell and degrade the laminin in the cell membrane. And when that happens, this cell gets perturbed, and it sends out danger signals. It sends out what are called cytokines, which cause a local inflammation, and that inflammation results in the opening of the tight junctions between the cells so the treponemes can go through and get into the bloodstream. And it does this in reverse to get out of the bloodstream to disseminate to other parts of the body. So let's go back to the natural history of syphilis. What causes the lesions. You know, the first thing you see is this ulcerative lesion called a chancre. What causes it? Well, when treponemes get there and begin to divide, they have lipoproteins. So this is a protein with a lipid or a fatty portion put onto it that are recognized by the innate immune system. And this is through a mechanism called toll-like receptor 2, which is not important for this purpose. But basically, they turn on the immune system by these lipoproteins. And then that leads to the sensitization of T lymphocytes and B lymphocytes and other cells, which then come to the site where all the treponemes are. And this shows you a lot of lymphocytes. All these things with dark nuclei, most of these, are lymphocytes. Later in infection, you'll see some macrophages come in. There is a macrophage right there. And they are responsible for clearing the primary infection, causing the lesion first and then clearing it. So let's talk about how they're cleared. This is a slide that shows the numbers of three particular entities. Treponema pallidum, the bacteria, which is a solid line. Lymphocytes, T lymphocytes, which are this dotted line, and then macrophages, which are the dashed line. So after infection, what happens is that the treponemes begin to multiply. They multiply and keep doing that until they reach their peak numbers. As they're multiplying, T lymphocytes are being sensitized because the immune response is being kicked into gear, and T cells keep coming in, and they reach their peak numbers about the same time the treponemes reach their numbers. And then following slightly behind that by several days are macrophages that come in. Macrophages are cells, specialized cells, whose job it is to run around and eat things that don't belong in the body. So they literally ingest them and destroy them. They break them down. And so you can see that as soon as the macrophages reach their peak numbers, the treponemes disappear, very clearly disappear. And then once the treponemes are gone, there's no need for the T cells and macrophages to hang around, and so they leave and the lesion heals. So to break this down into its component parts, here are the bacteria. Treponemes see those bacteria. T cells see the treponemes, and they become sensitized. They get turned on specifically to these organisms, antigens in these organisms, and they spit out something called gamma interferon. This is another cytokine, and it attracts macrophages to the area, and it turns those macrophages into really hungry, gobbling macrophages. They're activated. They're really, really quite good at ingesting things and killing them, much more so than just the quiet macrophages. At the same time, T pallidum is there, and they're being recognized by a different kind of lymphocyte called B lymphocytes. And B cells have receptors on them that are very specific for certain antigens. And so a B cell that recognizes an antigen on T pallidum will get turned on, and with some signals that come from the activated T cells will turn into antibody factories, and these are called plasma cells. These cells do nothing but pump out antibody to the particular antigen that they've recognized. So we now have activated macrophages. We have antibodies that are specific for T pallidum. And what those antibodies do is bind to the surface of the treponemes, and they make the treponemes more attractive to the activated macrophages. So the word opsonization comes from opsonos, which means to prepare the feast for. So it's like putting ketchup on a hot dog. It makes it much more palatable. So that's what happens. These treponemes have antibody on their surface. This macrophage says, oh boy, and it eats them and it kills them. And this is what clears literally billions of treponemes from the early lesions of syphilis. And so the target of that opsonic antibody, that kind of antibody, was kind of a holy grail, at least in my lab, for a while. And I have identified several targets of opsonic antibodies. So what this graph shows you, this is the percentage, this is an assay that's done in a culture dish. We put macrophages in the culture, we add serum, we put treponemes in, and we look to see whether or not the macrophages eat the treponemes. And as you see here, all these bright yellow, green dots are treponemes that are inside macrophages. If you put normal rabbit serum in, you get a little bit of phagocytosis, but not much. If you put in immune rabbit serum, you get much more phagocytosis. And then some of these other proteins that we've made antibodies to also are very good at opsonizing T. pallidum. And some that I'll talk about in the future are these green ones. That's TPR subfamily 1. That's a family of 4 proteins. TPRK we'll talk about later as well. And you can also note that 0751, which is that protein that I told you is important in Evasion, also is a target of opsonic antibodies. So the primary lesion is healed due to the host response, and so you've got a really good immune response going on. Well, how does secondary syphilis happen? If you've got such a good immune response, how does that happen? Obviously, the organisms have to get to the skin, right? So they have to disseminate. We've talked about how that happens. But why doesn't the immune response kill all those treponemes before they get there and before they can multiply to cause a lesion? Well, the key, we think, is evasion of the immune response. It's one of the things that many bacteria do, many pathogens do. The gonococcus, for example, is a classic example of somebody who can evade the immune response as well. But T. pallidum, it was discovered, also undergoes immune evasion. And this is called antigenic variation. The protein that is the target of the immune response is called the antigen, and that varies. So antigenic variation can be of several kinds. There's phase variation, where the antigen is either expressed or not expressed on the surface or anywhere in the organism, so it's made or not made by the bacterium. And this on-off mechanism is regulated by the organism, and there are some antigens in T. pallidum that have this phase variation on-off thing. The analogy I like to use for explaining the difference here is that if you're robbing a bank and you're wearing a red raincoat, then the police are going to be looking for someone with a red raincoat, right? So if you take that raincoat off, you can evade the police, and that's phase variation. That's the on-off. If you take that red raincoat off and you put on a yellow one, that's antigenic variation, which means that the raincoat's still being worn, but it's changed how it looks. And that's what the proteins do. That's what a particular protein does in T. pallidum. And that protein is TPRK. And of all of the 12 TPR proteins, TPRK is the most highly expressed. It's the one that's made the most on the surface of the organism. It's predicted to be in the outer membrane. It's in a place where the antibodies could bind to it. Some of the earliest immune responses in syphilis infection are antibodies to TPRK. And when you start looking at TPRK sequences from different strains, what you see is that there are some regions, shown here in brown, where the sequence is variable from one strain to another, and even within a strain, there are multiple sequences in these regions. The rest of it, the gray region, is all conserved. It's all the same in all strains. And during infection, antibodies that are made to TPRK are focused on these V regions, not on the rest of the protein. And this is just it's called a pileup. So it lists 5 different TPRK sequences, amino acid sequences. And all you need to know is that where it's black in the background, it's the same. Where it's white in the background, the sequences are different. So you can see here some of those variable regions and see that the sequence varies there, but not really in between those places. And if you do a structural prediction of this protein, you can see that these variable regions are on what's predicted to be on the outside of the bacteria. This part of the protein is in the membrane. It's embedded in the protein, in the membrane, and this part is between the outer membrane and the cell wall. But this is the outside, and so this is a place, obviously, where antibodies could easily bind an intact treponeme, right? And so the fact that antibodies are made to these V regions suggests that there's a lot of immune pressure that's put on the treponemes that are expressing V regions that the host has already seen. And so what happens is, well, here, let me I'm going to skip that slide and just show you immune selection. What happens is that if you infect with pink treponemes, for example, antibodies are made to the pink antigen, what's on the surface of the treponeme. But as these treponemes divide, they undergo the antigenic variation, and you have some treponemes that are made that have black or brown or blue on their surface rather than pink. But those that have the pink are opsonized. The antibody binds, macrophages are there, they gobble them up and kill them, and what's left are the variants. That's what's left. And so what happens in secondary syphilis is that the treponemes that are found in secondary lesions have a completely different V-region sequence in TPRK than those that cause the primary infection. We do this in a rabbit model so we can control what we're infecting with, and we can look to see what causes the secondary lesions. And we know that each lesion in secondary syphilis was seeded by a single treponeme. We know this because we can look at the TPRK V-region sequences and show that they're all the same in most cases, and they're different from the initial infecting sequence. So the fact that these organisms survived to cause the secondary lesions is because they escaped the immune response. So evolutionarily, why would you do this? Why would a bug do this? It's already caused a primary lesion. Why does it want to cause a secondary lesion? I kind of talk about my bacteria as if they're humans and have thoughts and plans, but it's a good way to think about what they do. So how many of you have seen this equation, R naught equals beta CD? Everybody? Most people? It has to do with the reproductive rate of an infection in a population, has to do with biological characteristics about how infectious this particular organism is, has to do with the number of susceptible contacts, and it has to do with the duration of the infectious stage. So if you think about why a bug might want to have a secondary stage, it prolongs the infectious period, a longer D, in the primary chancre, because these variants tend to appear in the primary chancre as well, but in much lower numbers. And then they disseminate to cause the secondary lesion. So it has a longer duration of infectiousness due to this antigenic variation. And it also increases the susceptible contact pool. So if you've been infected with one strain of T. pallidum and you've seen a certain set of TPRK V regions and have a really good immune response to those, if you infect with another strain or even the same strain expressing different TPRKs on it, then that can overcome the immunity that you've developed from the beginning, the first infection. And it permits patients to be reinfected as well. So we think this antigenic variation really can explain a lot about the natural history, but also some of the issues with regard to reinfection. And this work was all done by the secondary syphilis research, and the reinfection research was all done by graduate students in my lab. So how do we get to latent and tertiary syphilis? Well, these organisms, again, survive because they're variant. The immune system can't see them, so they avoid the immune response, and then they can pop back later after many years to cause disease. So what makes them come back later? We don't know. We don't have a good model for that. The rabbit's the best model for syphilis, but we don't really have a good model. And gets primary and secondary and latent syphilis, but it doesn't get tertiary syphilis, at least not in a lifetime that we can watch for. And so there are two factors that are involved. One is how good the host immune response is. And we know this from looking at people who have gotten organ transplants who happen to have syphilis. They see the syphilis can recur in those patients when the immune system is suppressed. We've seen through many, many years of HIV co-infection that when the immune system becomes depressed, that syphilis can again come back in some cases. And then, so those are host factors. The other has to do with strains of T. pallidum. Are there certain strains that are more likely or less likely to come back to cause progressive disease or to reappear. We know that there are differences among strains in the sequences in many of the TPR proteins, but also in other genes. So how can you look at this? Well, let's talk about neurosyphilis. It historically has been one of the most feared forms of syphilis because it can cause insanity, it can cause blindness, it can cause paralysis, it can cause death. And in the old literature, the clinical literature, there are references to neurotropic strains. So, for example, examples of a family where the husband, the mother, or the wife, the children all have syphilis, and they all develop neurosyphilis, which is not a very common event. Neurosyphilis is not a very common event. And so the thought was that there were some strains that were more likely to go into the central nervous system than others and to cause tertiary disease. And we did a study with Christina Mara in the rabbit model, and we did infect rabbits with different strains, different groups of rabbits with different strains of T. pallidum, and found that some strains were much more likely to be able to invade the central nervous system than others. The multiple strains can do it, but some seem to be more readily able to do that. So one way to look at that is to look at individual T. pallidum strains. And so the CDC group with Alan and Sheila have developed a published method for typing T. pallidum, and the method that they used is based on ARP and TPR, two different gene target areas. And then our group added a new gene target, TP0548. So an example type would be 14DC. So 14 is the number of repeats, of base pair repeats that you see in this particular gene. The D happens to be a particular pattern for the sequences when you cut the sequences of these genes. And then C, this last, where's my pointer? C has to do with the sequence in a new gene target, in this target 0548. So we have three different regions that we look at to type T. pallidum. And for example, a student in the lab looked at all of the strain types that we had samples for in Seattle from 2001 to 2010. And each strain type is a different color. And you can see that there was this one predominant strain type through 2007, and it pretty much disappeared. And then a new strain type came in. We don't know where this strain type came from. We don't know why this one went away. There are lots of ways we can speculate about that. But this is just an example of what you can see. And one of these, this 15DG, I believe it is, most of these were were MSM, but 15DG was a heterosexual couple, and they both had the same strain. So you can really tell, if you can get samples and do strain typing, you can do a lot of epidemiology with it, I think. But getting back to the question about clinical outcome and strain type, during this time, Christina Mara was doing a neurosyphilis study in Seattle, and of the patients that were enrolled in her study, 37% had neurosyphilis. And this was defined as having high white blood cells of greater than 20, because many of these people were HIV infected, and HIV alone can cause high white blood cells. So we raised the criteria to a higher number than is typical, to 20, or reactive CSF VDRLs. And in all the people in this study, there were 7 strain types that were seen. And of those who had strain type 14DF, half had neurosyphilis. And of those 39 people who had all of the other 6 strain types combined, 23% had neurosyphilis. So this was a significant association of this particular type with neurosyphilis. And if one could determine strain type ahead of time, you might be able to figure out which patients are at higher risk for neurosyphilis and then focus lumbar puncture on people who are at higher risk. This work was done in 2010, and since that time, she's continued to do this study, and it seems that the correlation with neurosyphilis is not based on the 14 or the D, but on the 548 type, the 0548 type. There are a number of types that you can find in patients with neurosyphilis, but type F seems to be the most predictive. So I think let's get to the question of reinfection. If you infect a rabbit with one strain of syphilis and let it sit infected for 3 months, and then you inject it intradermally with the same strain in a normal rabbit, that injection would cause the development of a chancre. In the rabbit that's been infected for 3 months, you don't get any lesions. You don't get any clinical signs of reinfection. But if you reinfect with a different strain, some of those different strains will cause chancres or superinfection. And Tara did some work that has led us to hypothesize we still need to do some more work. But it seems that the key in that reinfection is how different the TPRK variable regions are in your challenge strain compared to your first inoculum strain. So again, we have TPRK being, and this antigenic variation being central to the discussion about reinfection as well. So the last thing I want to talk about is vaccines. I think, as we talked earlier, those of you who were at the earlier discussion, I think we're never going to treat syphilis out of existence. Mary's here, and I hope that we can treat congenital syphilis out of existence, but I thought Mary was here. Oh, there she is. Yeah, I hope we can treat congenital syphilis out of existence, because by screening you theoretically should be able to do that. But I don't think we're going to get rid of it in adults. So we think a vaccine is the way to go. And what are the requirements for such a vaccine? You have to have a vaccine that can overcome this antigenic variation, which I've tried to convince you is so important in syphilis. We need to prevent development of the infectious lesions so that it can't be transmitted, and so it's also not a conduit for transmission or acquisition of HIV, which these lesions are. The other thing we need to do is to stop dissemination so that we don't have infection going to the fetus. We don't have infection going to the central nervous system, so you don't have the late sequelae of syphilis. So our proposal for doing that is to focus on inducing immune responses to conserved regions of antigens that are expressed on the surface of this bacteria. That should overcome the antigenic variation issue. We also want to induce antibodies that are opsonic, so that means they will bind to a surface of an intact organism and will make it more likely to be ingested and killed by macrophages. This will help from developing infectious lesions. And we also want to induce antibodies that inhibit the activity of this protein 0751 that's involved in dissemination. Caroline, I've got some data here. So this was immunization of rabbits with a conserved region of the TPR subfamily 1 proteins. There are 4 proteins there. They all share a conserved portion at the beginning of that protein. And so we immunized rabbits. We challenged them intradermally. In the unimmunized rabbits, you see this development of very ugly-looking lesions. A day or 2 after this, these lesions would ulcerate and would look very much like a primary chancre. They have tons of treponines in them. They would be infectious, were they in humans. This animal was immunized, and these black dots are ink marks showing right next to where we challenged them. So what you have here, although you do have some redness and some erythema, we couldn't identify treponines in these lesions by sticking a needle in and looking microscopically. We couldn't find treponines in them. They did not go on to ulcerate. And so in the unimmunized, 83% of challenge sites ulcerated compared to 8% in the immunized group. So we're pretty excited about this and are hoping to tweak the immunization schedule such that we can get even better protection. With regard to dissemination, Caroline has done studies where she has immunized with TP0751, and this sequence is 100% conserved in every strain of T. pallidum that we've been able to look at. She immunized the animals, challenged them on their backs at the skin site, just like I showed you in the last one. And then after a certain period of time, she took distant tissues, lymph nodes from distant places in the rabbits, and injected those into normal rabbits. And if those tissues, those lymph nodes, had viable treponemes in them, in other words, if the infection had disseminated, then the recipient animal would develop syphilis. The seroconvert would have treponeme. And if there were no treponemes in those lymph nodes, then the animals would not seroconvert. And here are the preliminary data with just a few animals. So people who got tissue from the unimmunized rabbits, all seroconverted, 2 out of 2 at day 17. Both of them had evidence of T. pallidum in tissues, and both of them developed clinical infection in the testes, which is where the injection was done. In the immunized animals, only 1 out of the 3 seroconverted, and that was at day 73. That was very late, suggesting there was a very, very, very small number of treponemes that were transferred in those lymph nodes. And they did not develop clinical manifestations, and there was no dark field evidence of T. pallidum. So very effective in stopping dissemination. So our plan for the vaccine is to have a cocktail of TPRs, the conserved regions of TPR, subfamily 1, and the conserved region of TPRK. And that should protect against chancre development, against transmission because you won't have chancres, against persistence because the organism should all be cleared there, against reinfection because again, you'll have antibodies on board to conserved regions, and you won't be able to overcome that by having different TPRK antigens. And if you prevent against chancres, you should also help to reduce HIV transmission and acquisition through the chancre. And by including 751, it should protect against dissemination, including congenital syphilis and neurosyphilis. So the take-home messages are, this is a smart bug. It's a really smart bug. It has co-evolved with humans over so long that it has jettisoned all the stuff it doesn't need, and it only has kept the genes that really, really matter. We know that an active immune response develops, and this immune response is very effective in clearing essentially all the bacteria locally. And that the clinical stages of syphilis are due to antigenic variation and therefore evasion of the immune response. That reinfection is possible, we think, because of this antigenic variation and, again, evasion of the existing immune response. And a successful vaccine will need to prevent both dissemination and transmission to be truly effective. So lastly, I just want to point out I want to thank the people who've worked on this work over the years. I mentioned Christine Amara. She's the one who has worked so carefully on neurosyphilis, and Caroline Cameron has done the beautiful work on 751 and dissemination. And the people in my lab, past and current lab, including my students, who've contributed to the work I've presented. So I'm happy to take any questions. Hi, Kim.
Published on the differences in opsonic antibody and HIV, and I was wondering if you could touch on that because that was very fascinating work, and I was wondering if you've looked at that in different stages of infection in people that have been reinfected.
Okay. So, yeah, that's a big, big question. There are lots of parts to that. So what Kim's referring to is a publication, a recent publication, in which the level of opsonization that occurs in CSF or serum, I can't remember, I think it was both. It was both, of people who are HIV infected versus those who are not HIV infected. And we found that using the same macrophages, so that was held steady, that variable was held steady, that people who were HIV infected had less opsonic capability than people who were not HIV infected. This was all done with the nickel strain as the target. So the question about specificity and whether it had to do with people who had been reinfected, I think is almost moot because the TPRK antigens would undoubtedly be different in the target nickel strain compared to the ones that these people were infected with. Mm-hmm. And during active infection, you do get opsonic antibody to a number of different proteins, But in TPRK, the antibodies are focused on the V regions. So a reinfection, I think the TPRK antibody is irrelevant to the assay that we just talked about, this opsonization assay in the example that you're giving. I don't know whether any of the samples that were tested were from people that had been reinfected, but I know that a high proportion of Christina's patients have been reinfected. But I don't know if they looked at the earlier versus the later serum sample. I know that their plans are to pair CSF and serum in reinfected patients with their first isolate and their second isolate. And I think there are about 3 such matched pairs for whom we have the isolates, but that work hasn't begun yet.
So that was just really fascinating recent work to me. And then thinking about how the use of ARVs or their starting CD4 count, how that affects.
Absolutely. I think much of what we've talked about with HIV-syphilis interaction over the years is going to be rendered moot in the setting where you can really get rid of the virus and keep it below detectable levels, and there's immune reconstitution going on. I think a lot of the odd things that we've seen in syphilis over the last 15, 20 years are going to be less important now, seen less frequently now that HIV, at least for the current moment, is under good control.
Thanks.
In some people, yeah. Ellen.
Thanks. That was a great talk. Just had some more questions about the vaccine work. You know, did you use a recombinant antigen? What was the time frame between immunizing and challenge? And, you know, how many more rabbits have you used? And if you could just elaborate a little bit.
Okay. We have done vaccine studies using a number of different recombinant antigens over the years, probably, I don't know, 12, 14 different recombinant antigens. And we used an adjuvant. The best results we got were with an adjuvant that is no longer made, unfortunately. An adjuvant is something you mix the antigens in order to immunize with because it stimulates an immune response. That vaccine, that adjuvant's no longer made, but we've been able to recreate it with component parts. And we've found a number of different proteins that induce some level of protection, partial protection. That is, we get fewer lesions that ulcerate, or we see fewer treponemes in aspirates from the challenge lesions. Those are all recombinant antigens that were not refolded. So when you make a recombinant antigen, you grow it in E. coli, you grow it in a bacterium. You have to then purify it from the E. coli. And because these are all membrane proteins, they're proteins that like to be in lipid and don't like to be in an aqueous form, they all form these things called inclusions. So they just form big gloms of protein. And in order to handle those, you have to denature the protein. And when you denature a protein, which is a long string, it's like a string of pop beads, when you denature them, they're just all long and strung out. When they're in their native form in the bacterium, they're all folded together like I showed you in that one structural prediction. And so all the things that we have done so far had been with denatured antigens. So clearly some of the targets of the immune response would have not been present in what we were immunizing with. So we're now immunizing with refolded proteins, recombinant proteins, that have the same, about the same combination of different kinds of structures in them that you would expect to find in the native protein. So it's as close to native as we can get it. In terms of how we immunized 5 injections at 3-week intervals in our rabbits. It's very expensive to do these studies, and we haven't had the time or the rabbits or the money to try, is 3 enough? Is 6 too many? Those kinds of studies. What if we waited a month instead of 3 weeks? All of those variables, it's very empiric. So you can only do what you can do. And so we have gotten good results with 5 at 3-week intervals, and that's what we'll continue with, at least and for the present time. Does that answer your question? Yeah.
Carter with Division of Scene. I'm going to bring us back to some basics.
Okay.
As a social scientist, so this has been fascinating. But I wonder if you could say a little bit about how penicillin interacts in all of this on the treatment side. I mean, just I know the treatment is typically a large dose. It's often 3 times at different intervals, at different phases. Sort of how does that interact, and also why penicillin still works for syphilis, and why we're not worried so much about.
I can't answer why penicillin still works.
Okay.
We do know that at least one of the penicillin binding proteins in T. pallidum has been looked at. It's called the T47 antigen. Has been looked at in a number of different strains, and we've not seen any variability at all in the sequence among many strains in that protein. So it suggests that those proteins are not able to vary. The other thing is that because most of those penicillin binding proteins are not on the surface, they're underneath the surface, there's not immune pressure on them to vary. And usually it's things that the immune response can attack that causes the pressure that leads to variation. So that's that. How does treatment affect developing immunity? Is that one of your questions? You wanted to know how penicillin interacts with this. So if the longer you have an infection, the more robust your immune response is going to be. If you treat really early, do you nip that immune response? Probably you do. This has been discussed in chlamydia as to why, if we do a lot of screening and we treat early, then people don't develop immunity, et cetera, et cetera. I think there's some truth to that. There's some truth to the fact that you don't get as mature an immune response if you treat any infection early than if you let it go longer. But obviously, we're not going to let it go longer. The other thing I would just say is that penicillin, benzosine penicillin, we know doesn't cross the blood-brain barrier. And so any treponemes that are in the central nervous system are not going to be touched by that. And does the presence of more organisms there continue to mature the immune response? I don't know the answer to that. Does it put the patient at risk? If they've got great immune systems, maybe not. If they don't have great immune systems, maybe. This is a long discussion, but that's it. Yeah.
Hi, good afternoon. Thank you so much for a clear and informative presentation. From your work, what do you anticipate it will take to move those vaccination studies from rabbits to humans, and how can we speed it up? Thanks.
Well, what it's going to take is money, and currently there are no vaccine studies being supported by NIH, syphilis vaccine studies being supported by NIH. This is an incredibly small field. There are probably 5 basic science labs in the United States who work on T. pallidum, and only less than a handful overseas. No, maybe in China. There are more in China now. But it's a small field, and our lab has been really the only one that has looked at protection induced by different antigens. There have been a couple of other studies over the years, over the last 25 years, but we're the only ones actively doing that. So it takes new people to come into the field, and in order to bring new people into the field, you need financial support because all of this work is expensive and it's supported by NIH. Are there any questions on the phone? Yes.
If you would like to ask a question over the phone at this time, please unmute your phone and press star 1.
Okay, Julia, you have a question. Oh, are you talking only about untreated syphilis causing reinfection? I'm not quite sure what that means. No, people who have been treated for syphilis, we know can be reinfected, if that's what you're asking. In our rabbit model, our rabbits were untreated. They were infected for 30 minutes, for 3 months, and then were challenged for reinfection. So I think it depends. I'm not quite sure exactly what you're getting at, but we know that people who've been treated can be reinfected again and again and again. And yes, it will be archived. The reason, someone asked me at the beginning whether this could happen right now. It says, sorry, this seems to have been, well, I don't know. It hasn't happened yet because there's one slide on there that I got that was unpublished data from Caroline Cameron, and I didn't want to say it could be saved and distributed until I got her permission. And I've emailed her, and as soon as I get her permission, if she says yes, the whole thing will be archived. If she says no, I'll take out that one single slide and we'll archive the rest. Any other, I don't see any other questions on the phone. Yeah, Mary.
Probably kind of naive, but I was really curious about, I thought the showing the strain variation in Seattle over time was really interesting, and then that historical discussion of neurotropic strains. So I wondered, from a basic science perspective, given, ocular syphilis is arising, what's known about neurotropic strains?
Well, we know that the strains that are type F, 0548 type F, seem to be more neurotropic than others, but that certainly doesn't exclude neurotropism in non-F strains. And what we don't know is whether it's that protein that we're looking at, that F protein that we're looking at, that really is responsible for neurotropism or not. We don't know that. We've tried to figure out how to ask that question and are not sure exactly how to test that. We don't know much more about neurotropism. I mean, we really don't. We really don't. So I don't think I answered your question, but we know very little, and no one else is looking at it besides Christy and Amara. So again, we need more people in this field and more brains working on this. Ours aren't big enough.
Sheila, this was fantastic. I want to really thank you. I think for the first time in 37 years, I have this much more understanding about syphilis thanks to you. Of course, I come from social science, so there's a reason for that. But you were so crystal clear, and it was fantastic. Thank you very much.
Thank you.
It is 2 o'clock. I think we have to bring this to closure formally, but I believe you have a little bit more time, so if people want to huddle and ask questions and talk with Sheila, that should be possible. Thank you all for being here, too.
Thank you.
Thank you for your participation on the phone. At this time, all parties may disconnect.